Game changer in oncology, tardigrade-derived protein shows 80% efficacy in shielding cells from radiation effects. Date, March 5, 2025. Events summary. Recent groundbreaking research conducted by a collaborative team from MIT Brigham and Women's Hospital and the University of Iowa has introduced a revolutionary approach to cancer treatment focusing on a damage-suppressing protein derived from tardigrades, also known as water bears. This protein, termed Dsup, has shown an impressive 80% efficacy in protecting healthy cells from radiation damage, which has significant implications for improving radiation therapy tolerability in cancer patients. Key findings and mechanism. Tardigrades are microscopic organisms famed for their extraordinary resilience, capable of surviving extreme environments, including exposure to high levels of radiation. The Dsup protein plays a crucial role in this resilience by binding to DNA and minimizing damage from radiation-induced strand breaks. Researchers aim to harness this natural mechanism to protect healthy cells during cancer treatment. Methodology. The research team developed a method to deliver messenger RNA mRNA encoding the Dsup protein to cells, prompting them to produce this protective protein before undergoing radiation therapy. In their experiments, the team injected nanoparticles containing Dsup mRNA into mice with oral cancer. Following radiation exposure, treated mice exhibited a 50% reduction in DNA damage compared to untreated controls, demonstrating the localized protective effects of the treatment. Mechanism of action. 1. Binding to DNA. The Dsup protein binds directly to DNA, effectively shielding it from radiation-induced damage. 2. Localized effect. The protective benefits of Dsup were restricted to the injection site, ensuring that tumor cells did not gain similar protection. 3. Temporary expression. The mRNA approach ensures that the protein is transiently expressed, minimizing long-term alterations to the cells. Research development timeline. Initial discovery. The unique properties of Dsup were identified as a potential tool for enhancing radiation therapy. Nanoparticle formulation. Polymer-lipid nanoparticles were developed to optimize the delivery of Dsup mRNA to target tissues. Testing phase. The approach was tested on mice, leading to significant findings regarding the reduction of DNA damage. Publication. The study was published in Nature Biomedical Engineering on February 26, 2025. Detailing the novel approach and its implications for cancer therapy. Common themes across sources. 1. Efficacy of Dsup. All sources highlight the 80% efficacy of Dsup in protecting healthy tissues from radiation damage. 2. Mechanism of action. The core mechanism involving the binding of Dsup to DNA and its ability to minimize damage is consistently described. 3. Potential applications. There are suggestions for broader applications beyond cancer therapy, including protecting astronauts from cosmic radiation and mitigating DNA damage from chemotherapy. Differences between sources. While all sources acknowledge the significance of the findings, they differ in focus. MIT News emphasizes the collaborative effort among institutions and details specific cancers that may benefit. The Scientist provides a technical breakdown of experimental methods and nanoparticle delivery systems. Genetic Engineering and Biotechnology News discusses implications for future research and the potential for scaling the treatment for human application. Future directions. The researchers plan to refine the Dsup protein to minimize immune responses, enhance delivery efficiency, and explore its applicability in various medical contexts. If successful in human trials, this innovative approach could revolutionize radiation therapy, allowing for more aggressive treatments while safeguarding healthy tissues, ultimately improving patient outcomes. Broader implications. The potential applications of the Dsup protein extend beyond cancer treatment. Researchers are optimistic about its use in protecting healthy tissues during chemotherapy and its potential role in space missions, where astronauts face exposure to harmful cosmic radiation. Conclusion. The integration of tardigrade-derived Dsup protein into cancer therapy represents a promising advancement in oncology with the potential to significantly reduce the side effects of radiation therapy. This research not only offers hope for improving the quality of life for cancer patients but also opens new avenues in the field of medical science, demonstrating the power of nature-inspired solutions in addressing complex challenges in healthcare. This innovative strategy could lead to a transformative change in the way radiation therapy is administered, making it safer and more effective for a larger number of patients. The commitment of researchers to further refine and develop this approach is an exciting prospect for the future of oncology and beyond. The research conducted by scientists at MIT Brigham and Women's Hospital and the University of Iowa has revealed the potential of a protein derived from tardigrades known as Dsup in revolutionizing cancer treatment by mitigating the damaging effects of radiation therapy. This study outlines a novel approach that employs messenger RNA mRNA to temporarily express this protective protein in healthy tissues, thereby enhancing the tolerability of radiation for cancer patients. Counterintuitive information. 1. Tardigrades as a model for human medicine. While tardigrades are often considered simple microscopic organisms, their extreme resilience to high radiation levels and other environmental stressors makes them a valuable model for complex human medical challenges. This highlights the unexpected potential of studying seemingly basic life forms to develop advanced therapeutic strategies. 2. Temporary mRNA expression versus genetic modification. The use of mRNA to express the Dsup protein temporarily contrasts with traditional gene therapy, which often involves permanent changes to the genome. This approach is counterintuitive as it suggests that short-term expression of a protective protein can be more advantageous and safer than long-term genetic modifications, which may pose risks of unintended consequences or immune responses. 3. Localized protection without tumor benefit. The study found that the protective effects of the Dsup protein were localized to the healthy tissues where the mRNA was injected, preventing any protective benefits to the tumor cells themselves. This is counterintuitive because one might assume that a broadly protective agent would also enhance the tolerance of tumor cells, possibly diminishing the efficacy of radiation therapy. The ability to shield healthy cells while ensuring that tumors remain vulnerable represents a significant step forward in cancer treatment. 4. Evolutionary biology informing medical innovation. The application of biological principles from an organism's evolutionary adaptations to human medicine underscores a counterintuitive shift in how we perceive the relevance of evolutionary biology. Rather than viewing evolution as a distant concept, this research illustrates its immediate applicability in solving pressing health issues, suggesting that insights derived from nature can lead to groundbreaking therapeutic advancements. 5. Potential beyond oncology. While the primary focus of the research is on cancer therapy, the implications of the Dsup protein extend to other fields, including protecting astronauts from cosmic radiation and reducing DNA damage from chemotherapy. This multifaceted applicability challenges the conventional notion that specific medical discoveries are limited to singular therapeutic contexts. 6. Dsup as a radioprotectant versus conventional drugs. The study emphasizes the limited options currently available to protect against radiation damage during cancer treatment, yet Dsup offers a novel mechanism that could surpass the efficacy of existing drugs. This suggests that leveraging naturally occurring proteins may provide more effective solutions compared to synthetic drugs, which often come with a host of side effects. 7. Safety of nanoparticle delivery systems. The research indicates that the combination of polymer and lipid nanoparticles for mRNA delivery is considered safer than direct protein injection, as the mRNA does not integrate into the genome. This challenges the perception that direct protein therapeutics are inherently safer and instead shows that using mRNA can offer a more controlled and reversible mechanism of action. 8. Broadening the scope of cancer research. The investigation into the Dsup protein as a protective agent against radiation damage is part of a broader push to find innovative methods of enhancing cancer treatment. This contrasts with the traditional focus on developing new chemotherapeutic agents or radiation techniques, suggesting that protective strategies can be equally important in the fight against cancer. Conclusion: the integration of tardigrade-derived Dsup protein into cancer therapy represents a promising advancement in oncology with the potential to significantly reduce the side effects of radiation therapy. The ability to protect healthy tissues while ensuring effective treatment of tumors exemplifies the innovative approaches that can arise from an interdisciplinary understanding of biology, engineering, and medicine. As research continues, it is expected that this methodology will not only enhance patient outcomes in oncology but also pave the way for applications in other fields, reinforcing the idea that nature can inform and inspire groundbreaking medical advancements. In the groundbreaking research on the tardigrade-derived protein Dsup, several key elements stand out that are often overlooked but are crucial for understanding the significance of this discovery in oncology and beyond. 1. Mechanism of action, beyond simple protection. While many reports emphasize Dsups' ability to bind to DNA and minimize radiation-induced damage, the specificity of this mechanism is significant. Dsup not only protects healthy cells but does so without affecting tumor cells. This selectivity is vital because it allows for effective radiation therapy without the risk of protecting malignant cells from treatment, which could compromise the efficacy of cancer therapies. The ability of Dsup to target only healthy tissues while leaving tumor cells vulnerable could fundamentally change how radiation therapy is applied in clinical settings. 2. mRNA delivery system, a novel approach. The use of messenger RNA mRNA to prompt cells to produce the Dsup protein is an innovative approach that merits attention. This strategy, which temporarily expresses the protective protein without integrating into the host genome, minimizes long-term risks associated with genetic modifications. This transient expression is particularly significant considering public concerns about gene therapy and its potential consequences. The focus on mRNA delivery, which is less likely to provoke an immune response compared to DNA constructs, marks a shift in therapeutic strategies that could be applicable across various medical fields, not just oncology. 3. Targeted nanoparticle delivery, engineering breakthrough. The development of polymer-lipid nanoparticles for efficient delivery of Dsup mRNA is a notable engineering achievement. This dual-component system enhances the delivery and uptake of mRNA, which is often a significant barrier in therapeutic applications. The researchers' method of screening various delivery vehicles to optimize this process is a testament to the importance of engineering in biomedical advancements. The implications of such targeted delivery systems extend beyond cancer therapy and could be adapted for various treatments requiring localized action without systemic side effects. 4. Broader applications beyond oncology. The potential applications of Dsup protein extend beyond cancer treatment. The researchers have hinted at its use in protecting astronauts from cosmic radiation and in mitigating DNA damage caused by chemotherapy. This versatility suggests that the findings could lead to advancements in fields such as space medicine and other medical therapies dealing with radiation exposure. The ability to harness natural protective mechanisms for diverse applications could open new avenues for research and treatment strategies in various medical disciplines. 5. Quality of life considerations. The emotional and psychological aspects of cancer treatment are often overshadowed by the focus on survival rates and clinical outcomes. The significant side effects of radiation therapy, which can lead to painful conditions like mouth sores and gastrointestinal issues, severely impact patients' quality of life. By reducing radiation-induced damage to healthy tissues, Dsup not only aims to enhance the efficacy of cancer treatments but also to improve the overall patient experience. The potential for reduced side effects could lead to higher treatment adherence and improved patient outcomes, a crucial consideration in oncology. 6. Future research directions and challenges. The researchers' plans to refine Dsup to minimize immune responses and enhance delivery efficiency are significant. The challenges of immunogenicity and the need for scalable production are often overlooked but are critical to translating this discovery from the laboratory to clinical use. Addressing these challenges may determine the success of Dsup-based therapies in human trials and their subsequent implementation in clinical oncology. 7. Interdisciplinary collaboration. The collaborative nature of this research involving experts from mechanical engineering, radiation oncology, and molecular biology highlights the importance of interdisciplinary approaches in solving complex medical challenges. This synergy not only brings together diverse expertise but also fosters innovation, suggesting that future breakthroughs in medicine may increasingly rely on such collaborations. Conclusion. The significance of the research surrounding the Dsup protein extends far beyond its immediate application in cancer therapy. The selective protection of healthy cells, the innovative use of mRNA delivery systems, the engineering of targeted nanoparticles, and the potential for broader applications all contribute to a transformative narrative in oncology and beyond. Moreover, the focus on enhancing patient quality of life and the collaborative nature of this research underscore a holistic approach to medical advancements. As the research progresses towards human trials, the implications of Dsup treatments could redefine how we approach radiation therapy and other areas of medicine, paving the way for a new era of patient-centered care. 1. Efficacy of Dsup and radiation protection. The research highlights the significant efficacy of the tardigrade-derived protein Dsup, which demonstrates an 80% effectiveness in shielding healthy cells from radiation damage during cancer treatment. This finding is crucial as it addresses a major challenge in oncology, protecting normal healthy tissues from the detrimental effects of radiation therapy, which can cause severe side effects like mouth sores, bleeding, and substantial pain. The ability of Dsup to reduce DNA strand breaks by binding to DNA represents a promising advancement in improving the tolerability of radiation therapy for cancer patients. 2. Innovative delivery method. The study introduces a groundbreaking method for delivering Tsup's protective effects through messenger RNA-mRNA encapsulated in polymer-lipid nanoparticles. This approach allows for the transient expression of the Dsup protein in targeted tissues, enhancing the safety profile compared to traditional DNA delivery methods that may integrate into the genome. The localized treatment not only minimizes the risk of systemic effects but also ensures that tumor cells remain susceptible to radiation. This innovative delivery system marks a significant step forward in the application of genetic engineering techniques in cancer therapy. 3. Potential for broader applications. The implications of this research extend beyond oncology. The Dsup protein and its delivery system may also be applicable in protecting astronauts from cosmic radiation, thus addressing the challenges of long-duration space missions. Additionally, there is potential for using Dsup to mitigate DNA damage from chemotherapy drugs, broadening its therapeutic applications. This versatility underscores the potential of leveraging biologically derived proteins for various medical and space exploration challenges. For future research directions and challenges, the research team plans to refine the Dsup protein to minimize immune responses and improve delivery efficiency for potential human applications. This includes developing a version of Dsup compatible with human physiology to reduce immunogenicity. Additionally, considerations regarding the manufacturing and scalability of this treatment are essential for transitioning from animal models to clinical trials. Addressing these challenges will be critical to realizing the full potential of Dsup as a revolutionary tool in enhancing cancer therapy and other medical applications. 1. The discovery of penicillin 1928. Alexander Fleming's accidental discovery of penicillin marked a turning point in medical history. After observing that the mold Penicillium notatum inhibited bacterial growth in his petri dishes, Fleming's research led to the development of penicillin as the first true antibiotic. This breakthrough revolutionized the treatment of bacterial infections, saving countless lives during and after World War II. The mass production of penicillin began in the early 1940s, providing a vital tool in combating infections that previously claimed many lives. The impact of penicillin extended beyond individual health, influencing public health policies and practices worldwide and laying the groundwork for the antibiotic era. 2. The development of insulin 1921. The isolation of insulin by Frederick Banting and Charles Best at the University of Toronto was a game-changer for diabetes management. Before insulin therapy, diabetes was a fatal disease and patients faced a grim prognosis. The introduction of insulin as a treatment transformed diabetes from a death sentence into a manageable chronic condition. Banting and Best's work, which involved extracting insulin from the pancreas of dogs, eventually led to the production of insulin for human use. This development not only saved lives but also spurred research into other hormonal treatments and the field of endocrinology. 3. The introduction of the polio vaccine 1955. The development of the polio vaccine by Jonas Salk represented a monumental achievement in public health. Polio, which caused widespread fear due to its potential to cause paralysis and death, was a significant health crisis in the early 20th century. Salk's inactivated poliovirus vaccine was tested and proved to be safe and effective in 1954, leading to its widespread distribution in 1955. The vaccination campaign dramatically reduced the incidence of polio, leading to the eventual near eradication of the disease in many parts of the world. This breakthrough not only saved thousands of lives but also set a precedent for vaccine development and public health initiatives aimed at controlling infectious diseases. Future outlook: the impact of tardigrade-derived protein on oncology and beyond. The recent breakthrough involving the tardigrade-derived protein Dsup, which has shown an 80% efficacy in shielding healthy cells from radiation damage, signifies a transformative moment in oncology and potentially other fields. As we project into the future, several key trends and developments are likely to unfold as this research advances. 1. Advancements in radiation therapy. The immediate future will likely see the integration of Dsup-based therapies into standard radiation treatment protocols. The ability to protect healthy tissue while allowing tumor cells to remain vulnerable to radiation could lead to enhanced treatment regimens. Oncologists may be able to prescribe higher radiation doses, improving tumor eradication rates without increasing the risk of collateral damage to healthy tissues. This could lead to better patient outcomes, reduced treatment times, and possibly even lower recurrence rates. Personalized oncology. The use of Dsup could pave the way for more personalized treatment plans where therapy is tailored based on individual patient responses, including genetic factors that influence how patients tolerate radiation. 2. Broader applications in cancer treatment. Beyond mere radiation therapy, the implications of Tsup's protective capabilities extend to chemotherapy. Given its potential to protect against DNA damage, Dsup may be developed to mitigate the side effects of chemotherapy, allowing for more aggressive treatment regimens without compromising patient quality of life. Combination therapies. The protein could be used in conjunction with other therapeutic modalities such as immunotherapy to enhance the overall effectiveness of cancer treatments while reducing adverse effects. 3. Regulatory and clinical pathways. As research progresses towards human trials, there will be significant focus on FDA approval process. Gaining regulatory approval will require rigorous clinical trials to assess safety, efficacy, and any potential long-term consequences of using mRNA technology in patients. Ethical considerations. The use of genetically engineered proteins raises ethical questions, especially regarding long-term genetic implications. Transparent discussions and guidelines will be essential as this technology moves toward widespread clinical use. 4. Technological innovations in delivery mechanisms. The nanoparticle delivery system for Dsup mRNA represents a significant leap in drug delivery technology. Future developments may include smart delivery systems. Innovations could lead to targeted delivery systems that release Dsup mRNA specifically in response to tumor markers, further enhancing safety and efficacy. Scalability in manufacturing. Advances in manufacturing processes for the polymer-lipid nanoparticles will be critical, ensuring that these therapies can be produced at scale to meet potential demand. 5. Applications beyond oncology. The versatility of Dsup may inspire applications far beyond oncology. Space exploration, the ability of tardigrades to withstand extreme radiation could translate into protective measures for astronauts, safeguarding them during long missions in deep space where exposure to cosmic radiation is a significant concern. Environmental and industrial applications. Dsup could be utilized in protecting biological systems in extreme environments, from industrial settings to bioremediation efforts where exposure to harmful radiation or chemicals is a risk. 6. Global health implications. If Dsup technology proves successful, it could have profound implications for global health. Access to treatment. By improving radiation therapy outcomes, the technology could contribute to increased survival rates in low-resource settings where cancer treatment options are limited. Healthcare costs. By reducing side effects and complications associated with radiation and chemotherapy, healthcare systems may see decreased hospitalization rates and overall treatment costs, making cancer care more accessible. 7. Research and development ecosystem. The Dsup discovery may catalyze a new wave of research into extremophile proteins and their applications. Future trends might include biomimicry in medicine. The success of Dsup could inspire further exploration of other extremophiles, leading to new discoveries in biomedicine that leverage nature's solutions to complex problems. Interdisciplinary collaborations. The convergence of disciplines such as molecular biology, materials science, and nanotechnology will be crucial in realizing the full potential of such innovations. Conclusion. The utilization of tardigrade-derived Dsup protein in oncology represents a significant breakthrough with the potential to reshape cancer treatment and beyond. As research progresses and this technology transitions into clinical settings, it holds the promise of not only improving patient outcomes in cancer therapy but also influencing a wide range of fields, from space exploration to environmental science. The next decade will be crucial as stakeholders, from researchers and clinicians to regulatory bodies and patients, navigate the opportunities and challenges presented by this innovative approach.